All right, here we go. We finally made it to the very last review video, topic 10. All 10 of these topics have covered everything that you could possibly see on the AP exam. Remember though that the exam is geared more towards data analysis and answering questions about experiments, and not so much on recall of information, but you can see in our practices, you do actually need to know a little bit of information. And then just being familiar with the background information actually can help you answer the questions about the experimental data that you see. So we're going to cover linked genes, biotechnology, and I eliminated chi-square. I was going to do chi-square, but I'm just going to go ahead and I'm going to trust what College Board said. And they said you don't need a calculator, but to have one handy. So I'm assuming they're not going to make you subtract, square, and then divide things. So I'm going to make the assumption chi-squared isn't on there. Um, if you are worried about chi-square, you can just Google chi-square videos and try to find something on YouTube, or I can send you something, but you're gonna have to email me to ask for that because I didn't want to overwhelm this email with anything that wasn't absolutely necessary. As you noticed on the email though, I did include all 10 topic videos and all 10 topic handouts for anyone that just couldn't get themselves to do it, but they just want that one last chance to get it done. And you could, the videos only range between like 10, some of them are 10, 20, 25, and I think there's like one that's 30 minutes. So it's definitely something that could still be done at this point, and it is highly recommended. So our first topic is linked genes. How can you tell if genes are linked on the same chromosome? So that's the first question. So genes that are linked together on the same chromosome tend to be inherited together unless they are divided due to crossing over. And this is just showing you examples of linked genes. Remember, you have black and vestigial. This is the wings of the fruit flies. And remember, if there's a plus sign, that's the wild type, which means dominant because Thomas Hunt Morgan just had to be different, and confuse us even more. But just remember, if you see a plus sign, because they might do that to y'all on Monday, if you see a plus sign, that's the wild type. Wild type means dominant. Just go with what you know about dominant and recessive. And remember, mutant means recessive. All right, so we need to understand the words parental type and recombinants. So if you have a parent that has a certain genotype and the offspring has that exact same genotype, well, that's considered a parental type. A recombinant will have one gene from one parent or one allele from one parent and one allele from another parent. All right, so if you look at this right here, this is parent one, heterozygous for yellow round. Here's parent two, see where my cursor is, homozygous recessive for green wrinkled. Any of the offspring that are heterozygous yellow round are a parental type. Anything that is homozygous green wrinkled is a parental type. So you see right here, here is a parental type, here is a parental type. Then you have these mixtures. So remember, one of the parents was green wrinkled, one of the parents was yellow round. Well, now you have a yellow wrinkled and a green round. That's what would make them recombinants. It's kind of like if you got your mom's hair and your dad's nose, you would be considered a recombinant, which most of us are anyways because we have so many genes. But we're just talking about simple pea plants here and simple fruit flies, because remember, fruit flies only have 4 chromosomes. Okay, so that's the difference between parental and recombinant. Okay, and you expect, because of the law of independent assortment, because of unlinked genes, that this would be a 1-to-1 to 1-to-1 ratio. If you had 1,000 peas from this cross, you would expect 250, 250, 250, 250, roughly, right? The recombinant frequency is the number of recombinants over total number of offspring. So if you calculate the recombinant frequency here, you have the number of recombinants over total number of offspring. So you would take yellow wrinkle, green round, add them together, divide by the total, and that would get you the recombination frequency. And you expect with unlinked genes for the recombination frequency to actually be around what? Yes, you are right. 50%. It's going to be close to 50% because it would a 50-50 chance that you're going to get a recombinant or a parental because of the law of independent assortment, right? Okay. So now we have a situation where we have two parents, gray, normal, heterozygous, black, vestigial. All right. Gray, normal, heterozygous, black, vestigial. So right here, you see, what is it saying? Gray body, normal wing, black, black body, vestigial. Okay, so this is body and wings. All right, so gray body, normal wings is dominant. Black body, vestigial wings is recessive. Okay, so now that makes sense. Okay, heterozygous gray, heterozygous vestigial, all right? Homozygous black, homozygous vestigial. Remember, if you see a plus sign, that means it's dominant. That's how you can tell this B plus and then this just B by itself. That is a heterozygous. VG plus and then VG by itself, that's heterozygous. Then homozygous recessive or mutant would be black vestigial. So here's a parental type, here's a parental type, here are your recombinants. You can just see by looking at this without even calculating recombination frequency, there's a disproportionate number of parental types versus recombinants. If you see a disproportionate number of parental types versus recombinants, you know those genes are linked on the same chromosome. So that first question, how can you tell genes are linked? There's a disproportionate number of parental types and recombinants among the offspring of two parents. Okay? They don't follow Mendel's law of independent assortment because there is no independent assortment if they're on the same chromosome. The only thing that accounts for recombinants, see this little number of recombinants here, is going to be crossing over. That off chance that crossing over occurred between those two genes and those two genes were actually broken apart. All right. So crossing over is going to explain how you have recombinants. And so anytime if you see, and there's always a multiple choice question about this. There has been a while back an FRQ about it. I don't really anticipate, I just don't know. College Board gets so creative with these questions. I would just know what parental and recombinant means, and then I would know, how do you know if there's a linked gene? It's when you have a disproportionate number of parental types. All right. So the farther apart away two genes are on a chromosome, the more likely they're going to get separated during crossing over because there's more spots between them for a breakage to occur. And so geneticists will create these linkage maps to just show how far apart two genes are from each other. One map unit means a 1% recombination frequency. Remember, recombination frequency is recombinants divided by total. And so that would be a really low number. As it approaches 50, once it gets to 50, you can pretty much assume they are either the farthest apart they can possibly be on a chromosome, or they are now located on two different chromosomes. Okay, so 50% recombination frequency, you most likely indicates they're on two different chromosomes. And that's really all I have for linked genes. Don't even look at this. I don't even know why that's on there. That's really all I have for linked genes. Non-disjunction and chromosome mutations. I don't have a slide for that, but just remember that non-disjunction is when chromosomes fail to separate during meiosis and can result in disorders like Down syndrome, where a person can have an extra chromosome. 4 types of chromosome mutations. This would be deletion, inversion, translocation, insertion, something like that. I don't anticipate that coming up, but just to name them again: translocation, deletion, insertion, and what did I say? Duplication, I think is what I said. All right. What is biotechnology? So now we're switching gears. So that was linked genes. I honestly, from what I just talked about, I would know parental type, recombinant type. How do you know you have a linked gene? I think that's all that could possibly show up. All right. Biotechnology is manipulating organisms and their components for the purpose of making useful products. So anytime I think of biotechnology, I think of, yes, we did the fun lab where we inserted the jellyfish gene into the bacteria and watched them glow, but that's not really biotechnology. That's like, genetic engineering, but it's not biotechnology because genetic engineering doesn't become biotechnology until you get a useful product from it. I mean, yes, the bacteria looked super cool and it's a learning um, activity for students, but we're not getting something. I mean, we want something we can sell, right? So it becomes biotechnology when we can take something from that organism and like sell it, especially to the medical community. So if we inserted a human insulin gene into the organism, sorry, I'm missing an N there, um, to produce insulin, mass produce insulin and sell it. We didn't do that. I mean, we could have ordered stuff to do that in our classroom to make insulin producing bacteria, but that would have been boring because you wouldn't have been able to see the insulin. So we would have been like, hey, here's a petri dish of bacteria. And then the next day we would have come in and I'd be like, yeah, they're making insulin. And we wouldn't have been able to prove it. The reason why the Piglo lab was so useful is because we could prove it because we could actually see it. Yay, and there's the pigs with the Piglo, with the bioluminescent jellyfish gene. Super cute. And you can make useful products like the rice with the beta carotene in it. Okay, someone just rang my doorbell. All right, so moving on, applications of gene cloning. You can use it for pest resistance, altering bacteria to clean up toxic waste, creating proteins that dissolve blood clots, and making things like human growth hormone. So drugs can be made or environmental cleanup products can be made. All right. We talked about PCR, polymerase chain reaction. Remember, polymerase chain reaction is just making billions of copies of DNA. And a lot of times this is involved in forensics. So if you find a little bit of DNA at a crime scene, you want to amplify it, make many copies so that you can do tests on it. And we did that. We did that online. I just love these online virtual labs. Not Labster so much. I'm so put off with Labster right now. It's unreal. But the one we did for PCR was good. Um, Remember the 3 steps: denaturation, heating up the DNA so that it splits apart, 2 primers, and then copying, copying, copying. Remember, if there's heating, then you need an enzyme that can withstand heat. So Taq polymerase is used as the polymerase that's going to create the new DNA strands. And that comes from a thermophilic heat-loving bacteria. And there's lots of errors in this, which is kind of scary. And the reason is because it just goes so fast, you know. All right, gel electrophoresis was our third biotechnology. It allows us to compare samples of DNA. So a lot of times you're going to do PCR first, then you're going to do electrophoresis. So electrophoresis is the one. Now College Board used to ask a lot of questions about transformation back in the day. I haven't seen it in a while, but I do see electrophoresis show up every couple of years. All right, so remember we have this power source with a cathode and an anode. We put our gel, our little jelly block. We put our buffer solution, and then we had wells that we loaded with DNA. Before we could load the wells with DNA, we put enzymes, restriction enzymes that were going to cut the DNA at very specific spots. So if 2 samples were identical, that enzyme is going to cut those samples at the exact same spots, leading to the exact same fragments in each one. And then you run them through the gel, and then at some point you push stop on the power source, and then you compare the banding pattern. So make your gel, add restriction enzyme to the DNA samples. So these little DNA samples in here. I did that before you came into the lab. I was so nervous it wasn't going to work. But some classes, it turned out really crazy, but some classes it worked really good. I just, if there's any contamination with these labs, it's just not going to work out very well. So you put the restriction enzyme into the DNA samples. The restriction enzyme chops it up, load the DNA in, turn on the current. So depending on where the restriction enzyme cut, you're going to end up with a bunch of DNA fragments that are different sizes. And so why that is significant is the smaller the fragment, the faster it's going to move through the gel, right? And that's because there's like these little sieves or these little pores. And if something is small, don't weigh very much, it's going to move through the pores a lot quicker. Okay, my son's playing with the doorbell and it's really annoying. all right, it's going to move a lot quicker. The bigger pieces are going to be a lot slower and they're going to stay behind. And so once you time it, now if you let an electrophoresis gel run forever, eventually all the DNA is just going to kind of fall off the edge. So you got to stop it at a very particular time and you see this banding pattern. And so here's what you can do. So let's say you have a blood stain from a crime scene, and who can forget our crime scene on Halloween? That was so much fun. Ugh, that was fun. So if you have a DNA sample from a crime scene and then you have 4 suspects. So let's say there was a struggle at this crime scene and the perpetrator's blood ended up at the crime scene. So what you're going to do is you're going to take the DNA from the perpetrator and you're going to try to match it. So you're going to chop up the DNA, all 5 of these with the same restriction enzyme. So if the DNA matches, the restriction enzyme is going to chop up the DNA the exact same way as the one it matches to, right? And then you compare banding patterns. So who is the guilty person? Yes, you are right. It is John. I show you this just because lithium bromide is carcinogenic, but it actually gets you the best banding patterns, and this is what's used in like really legit labs. In our lab, though, we use Carolina Blue, which is not carcinogenic, and you noticed with some of your gels, it just kind of smeared a little bit, but some of them actually turned out exactly the way the picture was. That was a really cool moment for me because after so many years of doing gel electrophoresis, this was actually the first year that it worked for somebody. I was about to give up gel electrophoresis for this year, and this is a common problem with all AP Bio teachers. It's not just me. But I have former colleagues that completely gave up doing gel electrophoresis, but I'm glad I didn't give up this year. Thanks, guys, for coming through for me with that. You renewed my faith in gel electrophoresis in the classroom. All right, here is our practice FRQ. Now, I could not find any FRQs that we have not already done that covered linked genes or biotechnology. I apologize, I searched, but I found an FRQ that's good, that is worth your while. So if you want to push pause, this would be I would say it'd be like an 8-minute FRQ. Time yourself 8 minutes. Push pause. Okay. So what we're going to do is go over the answer. All right. The data shows the growth of Saccharomyces cerevisiae. It's a yeast. So here's a yeast. You have a wild type. Remember, wild type is considered a dominant type. It's considered the dominant one that's normally found in nature. Mutant would be the ones that have the mutations. All right, so you see them, wild type and both mutants are grown with all amino acid present. Well, what does the plus sign mean? Oh, cool. The plus sign means the yeast strain is able to grow. The minus sign means the yeast strain is not able to grow. All right. The only way for the yeast to grow is if it has all of the necessary amino acids that it needs to grow. All right. And so what is the mutation? Well, it says that the synthesis of a particular amino acid can be prevented by a mutation, encoding an enzyme in the required pathway. All right. So you notice that your wild type is able to grow in any situation. You put that yeast wherever, and that yeast is going to do its thing. Why is that? Well, that's because it has all of the enzymes necessary to do its thing. It doesn't need these amino acids. It doesn't need everything except methionine. It doesn't need everything except leucine. It can grow wherever you put it. Put it on it's going to need some like nutrients like sugar and stuff like that. Just put it on a little sugar block, agar block. It can grow because it has the enzymes to do what it needs to do to make those amino acids, assuming that the right nutrients are actually kind of present, but not amino acids. So it can make its own amino acids. Then you notice mutant 1 and mutant 2 can grow when all amino acids are present because they don't have to do anything. They can just be lazy. They don't have to do anything. Mutant 1 can't grow here, can't grow here, but it can grow here. All amino acids except leucine. So what I can conclude is that mutant 1 has the enzyme where it's able to make leucine. And so it thrives. It's okay if you put all the amino acids except for leucine. the mutation isn't with the enzyme to make leucine, so this one's fine. However, if you put all amino acids except methionine, it can't make methionine. So obviously that's where the mutation is. And it's the opposite for mutant 2. It can grow when you have everything except methionine, so it must not be deficient in that enzyme. But it can't grow when there is no leucine, so it must be deficient in that enzyme. Hopefully that made sense. Hopefully you stopped the recording and you looked at the FRQ so that I didn't just confuse everyone. Identify the role of treatment 1. Treatment 1, all amino acids present. That is your control. That is the basis of comparison. That's all you would need to put. B, provide reasoning to explain how mutant 1 can grow on treatment 1, but cannot grow on treatment 3. Mutant 1 can grow on treatment 1 because it doesn't matter what mutation it has. If all the amino acids are present, it doesn't really matter. It doesn't really matter if it has a mutation in an enzyme, it's still able to grow. I hope that made sense. So it doesn't matter what enzyme it's deficient in, it's not needing to even make any amino acids because they're all present. All right. It cannot grow in treatment 3 because it must have a mutation in the enzyme needed for methionine. So that would be your reasoning. Yeast mate by fusing 2 haploid cells to make a diploid cell. Kind of like, humans do that. We have 2 haploid cells to make a diploid cell, and then it divides, and becomes a human. So it's kind of similar to what we do. In a second experiment, the researcher mates mutant 1 with mutant 2 to make a diploid cell. Predict what will happen in this column right here. And so this FRQ is on your paper, and hopefully you looked at it before listening to the answer. So what would happen is plus, 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 plus. Why is that? The reason that is because you have this one, which is able to make amino acids, which is able to make leucine, and you have this one, which is able to make methionine. If you have both of them together, you're good. Or the yeast. The yeast is good. It can make both, so it's not going to be deficient in anything. Hopefully that made sense. Here's the scoring guidelines if you want to pause the video just to read over what I just said. So there will be growth in all 4 cells. So you don't even have to give reasoning behind it. You just have to give a prediction. Yeah, when it says predict, you don't even have to explain yourself. All right, that's all I have for you today. That is our last review. I can't think of anything that I haven't gone over that I think could possibly be on your exam. Hopefully you got that FRQ I sent out yesterday. That FRQ I sent out yesterday is a really good FRQ to study. So hopefully, everyone will take the time to look at these videos, or at least some of these videos. I mean, anything can help. And if anyone has questions on what to expect on test day and what to do, please let me know now and not like Sunday night or Monday morning. That way I can help you. I think at this point, almost everyone has done the demo. There's a couple of people I actually still haven't heard from. I need to harass them a little bit. But I think we're good to go, especially a week after all the bugs are going to get worked out with College Board, and I don't think anyone should have any issues with their submissions. So let me know if you have any questions. I am going to give you one last practice FRQ tomorrow. It'll be a 15-minute one. I won't try to overwhelm you at the last minute, but so I'll give you a 15-minute FRQ. We'll go over it on Friday, and then just any last minute questions or anything like that we'll go over in our Zoom meeting, and then that'll be it for the year. All right, I will talk to you guys later.